22.2 Structure of prokaryotes, bacteria and archaea. By the end of this section, you will be able to do the following. Describe the basic structure of a typical prokaryote. Describe important differences in structure between archaea and bacteria. There are many differences between prokaryotic and eukaryotic cells. The name, prokaryote, suggests that prokaryotes are defined by exclusion. They are not eukaryotes, or organisms whose cells contain a nucleus and other internal membrane-bound organelles. However, all cells have four common structures. The plasma membrane, which functions as a barrier for the cell and separates the cell from its environment. The cytoplasm, a complex solution of organic molecules and salts inside the cell. A double-stranded DNA genome, the informational archive of the cell. And ribosomes, where protein synthesis takes place. Prokaryotes come in various shapes, but many fall into three categories: coxy, spherical, bacilli, rod-shaped, and spirally, spiral-shaped. Figure 22.9. Figure 22.9 Common prokaryotic cell types. Prokaryotes fall into three basic categories based on their shape, visualized here using scanning electron microscopy. A. Coxy, or spherical. A pair is shown, B. Bacilli, or rod-shaped, and C. Spirally, or spiral-shaped. Credit A. Modification of work by Janice Haney-Carr, Dr. Richard Facklam, CDC. Credit C. Modification of work by Dr. David Cox. Scale bar data from Matt Russell. The prokaryotic cell. Recall that prokaryotes are unicellular organisms that lack membrane-bound organelles or other internal membrane-bound structures. Figure 22.10. Their chromosome, usually single, consists of a piece of circular, double-stranded DNA located in an area of the cell called the nucleoid. Most prokaryotes have a cell wall outside the plasma membrane. The cell wall functions as a protective layer, and it is responsible for the organism's shape. Some bacterial species have a capsule outside the cell wall. The capsule enables the organism to attach to surfaces, protects it from dehydration and attack by phagocytic cells, and makes pathogens more resistant to our immune responses. Some species also have flagella, singular, flagellum, used for locomotion, and pili, singular, pilus, used for attachment to surfaces including the surfaces of other cells. Plasmids, which consist of extrachromosomal DNA, are also present in many species of bacteria and archaea. 542 Chapter 22, Prokaryotes, Bacteria and Archaea. Access for free at openstax.org. Figure 22.10 The features of a typical prokaryotic cell. Flagella, capsules, and pili are not found in all prokaryotes. Recall that prokaryotes are divided into two different domains, bacteria and archaea, which together with eukarya, comprise the three domains of life. Figure 22.11. Figure 22.11 The three domains of living organisms. Bacteria and archaea are both prokaryotes but differ enough to be placed in separate domains. An ancestor of modern archaea is believed to have given rise to eukarya, the third domain of life. Major groups of archaea and bacteria are shown. Characteristics of bacterial phyla are described in Figure 22.12 and Figure 22.13. Major bacterial phyla include the proteobacteria, the chlamydias, the spirochetes, the photosynthetic cyanobacteria, and the gram-positive bacteria. The proteobacteria are in turn subdivided into several classes, from the alpha to the epsilon proteobacteria. Eukaryotic mitochondria are thought to be the descendants of alpha proteobacteria, while eukaryotic chloroplasts are derived from cyanobacteria. Archaeal phyla are described in Figure 22.14. 22.2. Structure of prokaryotes, bacteria and archaea. 543 Figure 22.12 The proteobacteria. Phylum Proteobacteria is one of up to 52 bacteria phyla. Proteobacteria is further subdivided into 5 classes, alpha through epsilon. Credit, Rickettsia, modification of work by CDC. Credit, Spirillum modification of work by Wolfram Adlasnik. Credit, Vibrio cholera, modification of work by Janice Haney-Carr, CDC. Credit, Desulfovibrio vulgaris, modification of work by Graham Bradley. Credit, Campylobacter, modification of work by DeWood, Pooley, USDA, Ars, Emu. Scale bar data from Matt Russell, 544 Chapter 22, Prokaryotes. Bacteria and archaea access for free at openstax.org. Figure 22.13 Other bacterial phyla. Chlamydia, spirochetes, cyanobacteria, and gram-positive bacteria are described in this table. Note that bacterial shape is not phylum-dependent. Bacteria within a phylum may be coxy, rod-shaped, or spiral. Credit: Chlamydia trachomatis, modification of work by Dr. Lance Liotta Laboratory, NCI. Credit: Treponema pallidum, modification of work by Dr. David Cox, CDC. Credit: Firmidium, modification of work by USGS. Credit: Clostridium difficile, modification of work by Lois S. Wiggs, CDC. Scalebar data from Matt Russell, 22.2. Structure of prokaryotes. Bacteria and archaea 545. Figure 22.14 Archaeal phyla. Archaea are separated into 4 phyla: the Chorarchaeata, Euryarchaeata, Crenarchaeata, and Nanoarchaeata. Credit: Halobacterium, modification of work by NASA. Credit: Nanoarchaeatum equitans, modification of work by Carl O. Stetter. Credit: Chorarchaeata, modification of work by Office of Science of the U.S. Department of Energy. Scale bar data from Matt Russell. The plasma membrane of prokaryotes. The prokaryotic plasma membrane is a thin lipid bilayer, 6 to 8 nanometers, that completely surrounds the cell and separates the inside from the outside. Its selectively permeable nature keeps ions, proteins, and other molecules within the cell and prevents them from diffusing into the extracellular environment, while other molecules may move through the membrane. Recall that the general structure of a cell membrane is a phospholipid bilayer, composed of 2 layers of lipid molecules. In archaeal cell membranes, isoprene, phytanol, chains linked to glycerol replace the fatty acids linked to glycerol in bacterial membranes. Some archaeal membranes are lipid monolayers instead of bilayers. Figure 22.15. 546 Chapter 22. Prokaryotes. Bacteria and archaea access for free at openstax.org. Figure 22.15 Bacterial and archaeal phospholipids. Archaeal phospholipids differ from those found in bacteria and eukarya in 2 ways. First, they have branched phytanol side chains instead of linear ones. Second, an ether bond instead of an ester bond connects the lipid to the glycerol. The cell wall of prokaryotes. The cytoplasm of prokaryotic cells has a high concentration of dissolved solutes. Therefore, the osmotic pressure within the cell is relatively high. The cell wall is a protective layer that surrounds some cells and gives them shape and rigidity. It is located outside the cell membrane and prevents osmotic lysis, bursting due to increasing volume. The chemical composition of the cell wall varies between archaea and bacteria, and also varies between bacterial species. Bacterial cell walls contain peptidoglycan, composed of polysaccharide chains that are cross-linked by unusual peptides containing both L and D amino acids, including D-glutamic acid and D-alanine. Proteins normally have only L amino acids. As a consequence, many of our antibiotics work by mimicking D amino acids and therefore have specific effects on bacterial cell wall development. There are more than 100 different forms of peptidoglycan. S-layer, surface layer, proteins are also present on the outside of cell walls of both archaea and bacteria. Bacteria are divided into 2 major groups, gram-positive and gram-negative, based on their reaction to gram staining. Note that all gram-positive bacteria belong to one phylum. Bacteria in the other phyla, proteobacteria, chlamydias, spirochetes, cyanobacteria, and others, are gram-negative. The gram staining method is named after its inventor, Danish scientist Hans Christian Gram, 1853–1938. The different bacterial responses to the staining procedure are ultimately due to cell wall structure. Gram-positive organisms typically lack the outer membrane found in gram-negative organisms, figure 22.16. Up to 90% of the cell wall in gram-positive bacteria is composed of peptidoglycan, and most of the rest is composed of acidic substances called teichoic acids. Teichoic acids may be covalently linked to lipids in the plasma membrane to form lipoteichoic acids. Lipoteichoic acids anchor the cell wall to the cell membrane. Gram-negative bacteria have a relatively thin cell wall composed of a few layers of peptidoglycan, only 10% of the total cell wall, surrounded by an outer envelope containing lipopolysaccharides, LPS, and lipoproteins. This outer envelope is sometimes referred to as a second lipid bilayer. The chemistry of this outer envelope is very different, however, from that of the typical lipid bilayer that forms plasma membranes. 22.2. Structure of prokaryotes. Bacteria and archaea. 547 Visual connection. Figure 22.16 Cell walls in gram-positive and gram-negative bacteria. Bacteria are divided into two major groups, gram-positive and gram-negative. Both groups have a cell wall composed of peptidoglycan. In gram-positive bacteria, the wall is thick, whereas in gram-negative bacteria, the wall is thin. In gram-negative bacteria, the cell wall is surrounded by an outer membrane that contains lipopolysaccharides and lipoproteins. Porins are proteins in this cell membrane that allow substances to pass through the outer membrane of gram-negative bacteria. In gram-positive bacteria, lipoteichoic acid anchors the cell wall to the cell membrane. Credit. Modification of work by Franciscos 2 inches, Wikimedia Commons. Which of the following statements is true? A. Gram-positive bacteria have a single cell wall anchored to the cell membrane by lipoteichoic acid. B. Porins allow entry of substances into both gram-positive and gram-negative bacteria. C. The cell wall of gram-negative bacteria is thick, and the cell wall of gram-positive bacteria is thin. D. Gram-negative bacteria have a cell wall made of peptidoglycan, whereas gram-positive bacteria have a cell wall made of lipoteichoic acid. Archaean cell walls do not have peptidoglycan. There are 4 different types of archaean cell walls. One type is composed of pseudopeptidoglycan, which is similar to peptidoglycan in morphology but contains different sugars in the polysaccharide chain. The other 3 types of cell walls are composed of polysaccharides, glycoproteins, or pure protein. Other differences between bacteria and archaea are seen in Table 22.2. Note that features related to DNA replication, transcription and translation in archaea are similar to those seen in eukaryotes. Differences and similarities between bacteria and archaea. Structural characteristic. Bacteria. Archaea. Cell type. Prokaryotic cell morphology. Variable cell wall contains peptidoglycan, does not contain peptidoglycan. Cell membrane type. Lipid bilayer or lipid monolayer. Table 22.2548 Chapter 22. Prokaryotes. Bacteria and archaea. Access for free at openstax.org. Structural characteristic. Bacteria. Archaea. Plasma membrane. Lipids. Fatty acids. Glycerol ester. Phytanol. Glycerol ethers. Chromosome. Typically circular replication. Origin. Single multiple RNA polymerase. Single multiple initiator. tRNA. Formal methionine streptomycin. Inhibition. Sensitive resistant. Calvin cycle. Yes no. Table 22.2 Reproduction in prokaryotes is asexual and usually takes place by binary fission. Recall that the DNA of a prokaryote is a single, circular chromosome. Prokaryotes do not undergo mitosis. Instead, the chromosome is replicated and the 2 resulting copies separate from one another, due to the growth of the cell. The prokaryote, now enlarged, is pinched inward at its equator and the 2 resulting cells, which are clones, separate. Binary fission does not provide an opportunity for genetic recombination or genetic diversity, but prokaryotes can share genes by 3 other mechanisms. In transformation, the prokaryote takes in DNA shed by other prokaryotes into its environment. If a non-pathogenic bacterium takes up DNA for a toxin gene from a pathogen and incorporates the new DNA into its own chromosome, it too may become pathogenic. In transduction, bacteriophages, the viruses that infect bacteria, may move short pieces of chromosomal DNA from one bacterium to another. Transduction results in a recombinant organism. Archaea also have viruses that may translocate genetic material from one individual to another. In conjugation, DNA is transferred from one prokaryote to another by means of a pilus, which brings the organisms into contact with one another, and provides a channel for transfer of DNA. The DNA transferred can be in the form of a plasmid or as a composite molecule, containing both plasmid and chromosomal DNA. These 3 processes of DNA exchange are shown in Figure 22.17. Reproduction can be very rapid, a few minutes for some species. This short generation time coupled with mechanisms of genetic recombination and high rates of mutation result in the rapid evolution of prokaryotes, allowing them to respond to environmental changes, such as the introduction of an antibiotic, very quickly. 22.2. Structure of prokaryotes. Bacteria and archaea. 549 Figure 22.17 Gene transfer mechanisms in prokaryotes. There are three mechanisms by which prokaryotes can exchange DNA. In a transformation, the cell takes up prokaryotic DNA directly from the environment. The DNA may remain separate as plasmid DNA or be incorporated into the host genome. In B. Transduction, a bacteriophage injects DNA into the cell that contains a small fragment of DNA from a different prokaryote. In C. Conjugation, DNA is transferred from one cell to another via a mating bridge, or pilus, that connects the two cells after the sex pilus draws the two bacteria close enough to form the bridge. Evolution connection. The evolution of prokaryotes. How do scientists answer questions about the evolution of prokaryotes? Unlike with animals, artifacts in the fossil record of prokaryotes offer very little information. Fossils of ancient prokaryotes look like tiny bubbles in rock. Some scientists turn to genetics and to the principle of the molecular clock, which holds that the more recently two species have diverged, the more similar their genes, and thus proteins, will be. Conversely, species that diverged long ago will have more genes that are dissimilar. Scientists at the NASA Astrobiology Institute and at the European Molecular Biology Laboratory collaborated to analyze the molecular evolution of 32 specific proteins common to 72 species of prokaryotes. The model they derived from their data indicates that three important groups of bacteria, actinobacteria, Deinococcus, and cyanobacteria, collectively called pterobacteria by the authors, were the first to colonize land. Actinobacteria are a group of very common gram-positive bacteria that produce branch structures like fungal mycelia, and include species important in decomposition of organic wastes. You will recall that Deinococcus is a genus of bacterium that is highly resistant to ionizing radiation. It has a thick peptidoglycan layer in addition to a second external membrane, so it has features of both gram-positive and gram-negative bacteria. Cyanobacteria are photosynthesizers, and were probably responsible for the production of oxygen on the ancient Earth. The timelines of divergence suggest that bacteria, members of the domain Bacteria, diverged from common ancestral species between 2.5 and 3.2 billion years ago, whereas the archaea diverged earlier, between 3.1 and 4.1 billion years ago. Eukarya later diverged from the Archean line. The work further suggests that stromatolites that formed prior to the advent of cyanobacteria, about 2.6 billion years ago, photosynthesized in an anoxic environment, and that because of the modifications of the pterobacteria for land, resistance to drying and the possession of compounds that protect the organism from excess light, photosynthesis using oxygen may be closely linked to adaptations to survive on land. Batistuzzi, Fu, Feijao, A., and Hedges, S.B. A Genomic Timescale of Prokaryote Evolution. Insights into the Origin of Methanogenesis, Phototrophy, and the Colonization of Land. Biomed Central. Evolutionary Biology 4, 2004, 44, doi. 10. 1186-1471 to 2148 4-44.